In biology, a species is one of the basic units of biological classification and a taxonomic rank. A species is often defined as the largest group of organisms where two individuals are capable of reproducing fertile offspring, typically using sexual reproduction. While in many cases this definition is adequate, the difficulty of defining species is known as the species problem. Differing measures are often used, such as similarity of DNA, morphology, or ecological niche. Presence of specific locally adapted traits may further subdivide species into infraspecific taxa, such as subspecies. Species hypothesized to have the same ancestors are placed in one genus, based on similarities. The similarity of species is judged based on comparison of physical attributes, and where available, the DNA sequences. All species are given a two-part name, a binomial name, or just binomial. The first part of a binomial is the generic name, the genus to which the species belongs. The second part is either called the specific name or the specific epithet. For example, boa constrictor is one of 4 species of the boa genus. While the genus gets capitalized, the specific epithet does not. The binomial is written in italics when printed and underlined when handwritten. A usable definition of the word species and reliable methods of identifying particular species are essential for stating and testing biological theories and for measuring biodiversity. Though other taxonomic levels such as families may be considered in broad-scale studies, extinct species known only from fossils are generally difficult to assign precise taxonomic rankings, which is why higher taxonomic levels such as families are often used for fossil-based studies. The total number of non-bacterial and non-archaeal species in the world has been estimated at 8.7 million, with previous estimates ranging from 2 million to 100 million. History and development of the concept. In the earliest works of science, a species was simply an individual organism that represented a group of similar or nearly identical organisms. No other relationships beyond that group were implied. Aristotle used the words genus and species to mean generic and specific categories. Aristotle and other pre-Darwinian scientists took the species to be distinct and unchanging, with an essence, like the chemical elements. When early observers began to develop systems of organization for living things, they began to place formally isolated species into a context. Many of these early delineation schemes would now be considered whimsical, and these included consanguinity based on color or behavior. John Ray, an English naturalist, was the first to give a biological definition of the term species. In the 18th century, Swedish scientist Carl Linnaeus classified organisms according to shared physical characteristics, and not simply based upon differences. He has also established the idea of a taxonomic hierarchy of classification based upon observable characteristics and intended to reflect natural relationships. At the time, however, it was still widely believed that there was no organic connection between species, no matter how similar they appeared. This view was influenced by European scholarly and religious education at the time, which held that the categories of life are dictated by God, in a hierarchical scheme. Although there are always differences between individual organisms, Linnaeus strove to identify individual organisms that were exemplary of the species, and considered other non-exemplary organisms to be deviant and imperfect. By the 19th century, most naturalists understood that species could change form over time, and that the history of the planet provided enough time for major changes. Jean-Baptiste Lamarck, in his 1809 zoological philosophy, offered one of the first logical arguments against creationism. The new emphasis was on determining how a species could change over time. Lamarck suggested that an organism could pass on an acquired trait to its offspring. With the acceptance of the natural selection idea of Charles Darwin in the 1860s, however, Lamarck's view of goal-oriented evolution, also known as a teleological process, was eclipsed. Recent interest in inheritance of acquired characteristics centers around epigenetic processes that do not affect DNA sequences, but instead alter expression in an inheritable manner. Thus, neo-Lamarckism, as it is sometimes termed, is not a challenge to the theory of evolution by natural selection. Charles Darwin and Alfred Wallace provided what scientists now consider as the most powerful and compelling theory of evolution. Darwin argued that it was populations that evolved, not individuals. His argument relied on a radical shift in perspective from that of Linnaeus. Rather than defining species in ideal terms, Darwin considered variation among individuals to be natural. He further argued that variation, far from being problematic, actually provides the explanation for the existence of distinct species. Darwin's work drew on Thomas Malthus's insight that the rate of growth of a biological population will always outpace the rate of growth of the resources in the environment, such as the food supply. As a result, Darwin argued, not all the members of a population will be able to survive and reproduce. Those that did will, on average, be the ones possessing variations, however slight, that make them slightly better adapted to the environment. If these variable traits are heritable, then the offspring of the survivors will also possess them. Thus, over many generations, adaptive variations will accumulate in the population, while counter-adaptive traits will tend to be eliminated. Whether a variation is adaptive or non-adaptive depends on the environment. Different environments favor different traits. Since the environment effectively selects which organisms live to reproduce, it is the environment that selects the traits to be passed on. This is the theory of evolution by natural selection. In this model, the length of a giraffe's neck would be explained by positing that proto-giraffes with longer necks would have had a significant reproductive advantage to those with shorter necks. Over many generations, the entire population would be a species of long-necked animals. In 1859, when Darwin published his theory of natural selection, the mechanism behind the inheritance of individual traits was unknown. Although Darwin made some speculations on how traits are inherited, his theory relies only on the fact that inheritable traits exist and are variable. Although Gregor Mendel's paper on genetics was published in 1866, its significance was not recognized. It was not until 1900 that his work was rediscovered by Hugo de Vries, Carl Correns and Erich von Schirmacher, who realized that the inheritable traits in Darwin's theory are genes. The theory of the evolution of species through natural selection has two important implications for discussions of species consequences that fundamentally challenge the assumptions behind Linnaeus taxonomy. First, it suggests that species are not dissimilar, they may actually be related. Some students of Darwin argue that all species are descended from a common ancestor. Second, it supposes that species are not homogeneous, fixed, permanent things. Members of a species are all different, and over time species change. This suggests that species do not have any clear boundaries but are rather momentary statistical effects of constantly changing gene frequencies. One may still use Linnaeus taxonomy to identify individual plants and animals, but one can no longer think of species as independent and immutable. The rise of a new species from a parental line is called speciation. There is no clear line demarcating the ancestral species from the descendant species. Although the current scientific understanding of species suggests that there is no rigorous and comprehensive way to distinguish between different species in all cases, biologists continue to seek concrete ways to operationalize the idea. One of the most popular biological definitions of species is in terms of reproductive isolation. If two creatures cannot reproduce to produce fertile offspring of both sexes, then they are in different species. This definition captures a number of intuitive species boundaries, but it remains imperfect. It has nothing to say about species that reproduce asexually, for example, and it is very difficult to apply to extinct species. Moreover, boundaries between species are often fuzzy. There are examples where members of one population can produce fertile offspring of both sexes with a second population, and members of the second population can produce fertile offspring of both sexes with members of a third population. But members of the first and third population cannot produce fertile offspring, or can only produce fertile offspring of the homozygous sex. Consequently, some people reject this definition of a species. Richard Dawkins defines two organisms as conspecific if and only if they have the same number of chromosomes, and for each chromosome, both organisms have the same number of nucleotides. However, most taxonomists would disagree. For example, in many amphibians, most notably in New Zealand's Leopelma frogs, the genome consists of core chromosomes that are mostly invariable and accessory chromosomes, of which exist a number of possible combinations. Even though the chromosome numbers are highly variable between populations, these can interbreed successfully and form a single evolutionary unit. In plants, polyploidy is extremely commonplace with few restrictions on interbreeding, as individuals with an odd number of chromosome sets are usually sterile, depending on the actual number of chromosome sets present. This results in the odd situation where some individuals of the same evolutionary unit can interbreed with certain others and some cannot, with all populations being eventually linked as to form a common gene pool. The classification of species has been profoundly affected by technological advances that have allowed researchers to determine relatedness based on molecular markers. Starting with the comparatively crude blood plasma precipitation assays in the mid-20th century to Charles Sibley's DNA hybridization studies in the 1970s leading to DNA sequencing techniques. The results of these techniques caused revolutionary changes in the higher taxonomic categories, resulting in the reordering of many branches of the phylogenetic tree. For taxonomic categories below genera, the results have been mixed so far. The pace of evolutionary change on the molecular level is rather slow, yielding clear differences only after considerable periods of reproductive separation. DNA hybridization results have led to misleading conclusions, the Pomerine skewer, Great Skewer phenomenon being a famous example. Turtles have been determined to evolve with just 1/8 of the speed of other reptiles on the molecular level, and the rate of molecular evolution in albatrosses is half of what is found in the rather closely related storm petrels, both being within the Procellaria forms. The hybridization technique is now obsolete and is replaced by more reliable computational approaches for sequence comparison. Molecular taxonomy is not directly based on the evolutionary processes, but rather on the overall change brought upon by these processes. The processes that lead to the generation and maintenance of variation, such as mutation, crossover and selection, are not uniform. DNA is only extremely rarely a direct target of natural selection rather than changes in the DNA sequence enduring over generations being a result of the latter. For example, silent transition transversion combinations would alter the melting point of the DNA sequence, but not the sequence of the encoded proteins, and thus are a possible example where, for example in microorganisms, a mutation confers a change in fitness all by itself. Biologists' working definition, a usable definition of the word species, and reliable methods of identifying particular species is essential for stating and testing biological theories and for measuring biodiversity. Traditionally, multiple examples of a proposed species must be studied for unifying characters before it can be regarded as a species. Some biologists may view species as statistical phenomena, as opposed to the traditional idea, with a species seen as a class of organisms. In that case, a species is defined as a separately evolving lineage that forms a single gene pool, although properties such as DNA sequences and morphology are used to help separate closely related lineages. This definition has fuzzy boundaries. However, the exact definition of the term species is still controversial, particularly in prokaryotes, and this is called the species problem. Biologists have proposed a range of more precise definitions, but the definition used is a pragmatic choice that depends on the particularities of the species of concern. Common names and species are commonly used names for plant and animal taxa sometimes correspond to species. For example, lion, walrus, and camphor tree each refers to a species. In other cases, common names do not. For example, deer refers to a family of 34 species, including eld's deer, red deer and elk. The last two species were once considered a single species, illustrating how species boundaries may change with increased scientific knowledge. Placement within genera. Ideally, a species is given a formal, scientific name, although in practice there are very many unnamed species. When a species is named, it is placed within a genus. From a scientific point of view, this can be regarded as a hypothesis that the species is more closely related to other species within its genus than to species of other genera. Species and genus are usually defined as part of a larger taxonomic hierarchy. The best-known taxonomic ranks are, in order: life, domain, kingdom, phylum, class, order, family, genus, and species. This assignment to a genus is not immutable. Later a different taxonomist may assign it to a different genus, in which case the name will also change. In biological nomenclature, the name for a species is a two-part name, treated as Latin, although roots from any language can be used as well as names of locales or individuals. The generic name is listed first, followed by a second term. The terminology used for the second term differs between zoological and botanical nomenclature. In zoological nomenclature, the second part of the name can be called the specific name or the specific epithet. For example, gray wolves belong to the species Canis lupus, coyotes to Canis latrans, golden jackals to Canis aureus, etc. And all of those belong to the genus Canis. For the gray wolf, the genus name is Canis, the specific name or specific epithet is lupus, and the bonomen, the name of the species, is Canis lupus. In botanical nomenclature, the second part of the name can only be called the specific epithet. The specific name in botany is always the combination of genus name and specific epithet. For example, the species commonly known as the longleaf pine is Pinus palustris, the genus name is Pinus, the specific epithet is palustris. The specific name is Pinus palustris. This binomial naming convention, later formalized in the biological codes of nomenclature, was first used by Leonhard Fuchs and introduced as the standard by Carolus Linnaeus in his 1753 Species Plantarum. Abbreviated names, books and articles sometimes intentionally do not identify species fully and use the abbreviation SP in the singular or SPP in the plural in place of the specific name or epithet. This commonly occurs in the following situations. The authors are confident that some individuals belong to a particular genus but are not sure to which exact species they belong. This is particularly common in paleontology. The authors use SPP as a short way of saying that something applies to many species within a genus, but do not wish to say that it applies to all species within that genus. If scientists mean that something applies to all species within a genus, they use the genus name without the specific name or epithet. Sometimes, the aforementioned plural is rendered as SPS, which may lead to confusion with SSP, this one standing for subspecies instead. In books and articles, the names of genera and species are usually printed in italics. Abbreviations such as SP, SPP, SPS, SSP, SUBSP, etc. should not be italicized. Better source needed. Identification codes. Various codes have been devised for identifying particular species. For example, National Center for Biotechnology Information employs a numeric taxid or taxonomy identifier, a stable unique identifier, e.g., the taxid of H. sapiens is 9606. Kyoto Encyclopedia of Genes and Genomes employs a 3 or 4 letter code for a limited number of organisms. In this code, for example, sapiens is simply HSA. Uniport employs an organism mnemonic of not more than 5 alphanumeric characters, e.g., human for H. sapiens. Integrated Taxonomic Information System provides a unique number for each species.